I'd just like to go over some of the material from week 2, the cancer genetics lecture. Most of this lecture concerns the multi-hit hypothesis of cancer formation, and then there's a key example of cancer, such as colon cancer, which was worked through in more detail with reference to specific genetic mutations, specific genes that are involved in the process. So we kind of looked at some of the data that describes cancer formation. Basically, it takes many years to form cancers, as we know, and typically it's a disease of old people, but there's a lot more to it than that. So basically, there's been accumulating evidence for a long time before the genetic factors were known that the development of cancer involved multiple genetic changes. That there's not a single genetic change that will give you cancer, but there's an accumulation of genetic changes within the same cell that will give you cancer. And during week 14, I showed that video from the Howard Hughes Medical Institute, and that they talked about the multi-step hypothesis and clonal expansion of cells that acquire mutations. So there's a really nice animation and some background information in that video from week 14. So we started to focus on cancers of the epithelial cells because colon cancer is a cancer of epithelial cells. And we just looked at the rate of turnover and some of the characteristics of these cells. And also, I mean, it's not an overly strict rule, but cancers typically originate in proliferating cells, so the colon cells are constantly turning over because because of the nature of their function. So, and I guess one of the reasons I focused on colon cancer, or the textbooks focus on colon cancer, is because it's an easy disease to track because you can inspect colons and you can look at colons. It's a fairly non-invasive technique. It's not like you've got to take like it's very difficult to see what's happening inside lungs and inside livers and things, but it's very easy with modern technology to observe changes in the lining of the colon. So it's a disease that can be tracked over time and in a fairly non-invasive kind of way. So they're looking at the lining of the colon, or has these villi, these little protrusions which have very thin membranes to absorb nutrients and all that kind of stuff. And there's lots of histopathology records that show the development of a malignant growth within this tissue. These sort of growths are referred to in their initial stages as polyps, and these are these little growths that are attached to the wall of the colon, and typically these are benign, but from these polyps other mutations can occur which can become malignant. So like here's one of these villi we were talking about, and here's just some sort of histopathology samples that show a slight aberration and how it grows and gets larger over time. So I guess given that we've got a succession of changes occurring from these polyps, which then start to have these outgrowths and they start to get more malignant, as you've got the progression of the development of the cancer, at a molecular level, you've got a progression of genetic changes. And this is this idea of a multi-step of a series of events, successive genetic alterations within the cell that is causing these cancers. So again, just looking in this cartoon here, we go from normal kind of phenotype, and then a series of severe changes up until we have a cancerous state. And this might take 10, 15 years to progress from normal cells to the cancerous cells. So at various stages during the progress of the disease, these polyps and can be looked at through colonoscopies and things like that. So I guess we cover the idea that it might not be that there might be an alternative hypothesis of how the cancers develop, but when you look at the data in a bit more detail, it's pretty much a multi-step hypothesis and a continuation of one set of mutations which sort of establish a new colony of cells, and then another mutation occurs within that colony, and so forth. So it's this idea of a succession of phenotypes is the multi-step hypothesis, and here's a bit of evidence for that, showing you've got a polyp growing at some stage here, and then from these cells from which the polyp has developed, the cancer is growing out of these cells. So within the normal cells, there's been a genetic change leading to the formation of the polyp, and within those cells, there's been a second genetic change, at least a second genetic change, that's led to a more malignant phenotype. There's probably a succession of genetic changes that occur. Also, if you remove the polyps from patients, they have a much less chance of developing more malignant phenotypes than if you leave the polyps intact, clearly indicating that the malignant phenotypes are coming from the polyps. And that's another indication that these mutations are occurring in a single cell line. And there's also good genetic evidence that's come out of some familiar studies which show that that certain genetic mutations are prone to developing these polyps, and you can study the development of cancer in these patients. So looking at the course of development of cancers in general, we looked at the colon cancer in a little bit of detail, but this is true for many other cancers as well. So this idea of the multi-hit hypothesis, even though it's well documented in some cancers and not so well documented in others, it's thought that it's the same general process of this multiple genetic changes occurring, leading to all these different cancers. But it's different factors that have been genetically altered in different cancers, clearly. So people who have been studying colon cancer, I won't go into the historical sort of timeline of discovery of these factors, but effectively, if you're working through the progression from normal cells to invasive cancerous cells, they've picked up a range of different genetic factors that are involved. So initially they picked up something on chromosome 5 that was involved, and then they picked up something in different regions that were known. Later studies showed that the region of chromosome 5 that was important in the early stages of development was a loss of a gene called APC. And there's also some epigenetic changes such as hypomethylation that occur, and there's also other genetic changes have been better characterized during the development of the disease. So this will only get more and more well characterized over time. So typically this introduces some of the key players that we know, and some key concepts such as oncogenes and tumor suppressors, which are the genetic factors that are involved in the progression of the disease. So typically there's going to be a couple of oncogenes involved in tumor progression, some mutations in oncogenes, and there's also going to be some inactivation of tumor suppressor genes involved. And I guess a specific example of colon cancer is that here we have the progression of the disease as observed, within a colon. This is a person's colon here, a normal colon without polyps, and then the progression of a growth during many years. And then as the development of the cancer proceeds, they've picked up various genetic changes that occur by taking biopsies of patient samples and investigating genetically what's happening. They've picked up these different genetic changes that are occurring. So in this way, they've been able to fairly well characterize key multiple events. So this is that multi-step hypothesis. They picked up the multiple steps that are required to progress from normal to cancerous for this particular cancer. And they've done it for other cancers as well, but we don't go into any detail with those. So it's not a linear progression from normal to mutated. Okay, so each person has a distinct series of genetic events that occur, but the APC gene is particularly important for a mutation that occurs early on in the progression of the disease. And there's lots of other key players. There's tyrosine kinases, there's phosphoinositol kinases, there's RAFs and RASs. There's a whole bunch of oncogenes and key players that can be mutated. So one particular patient might have APC mutated, might have RAS mutated, might have this unidentified factor mutated, and it might have some other things. Whereas another patient may have a RAS mutation and a P53 mutation, as well as the APC mutation. There's the whole range of different genetic events that lead to the same outcome, which is these cells with an increased growth potential, with an increased rate of proliferation over normal cells. And then we go through to look at the end game, which is the metastasis, the spread of the cancer from the initial site through to other tissues. And I'll just quickly skip through this idea of clonal selection, which was covered in the video from week 14, as well as here. It's just indicating the multiple steps, the multiple mutations that occur within the same cohort of cells, which gives them increasingly advantageous growth characteristics. So this idea of metastasis is what makes the cancers really dangerous, in the fact that they can then move from their initial site, where they're not causing a great deal of harm, to another site in the body to cause a great deal of harm, and effectively inactivate a tissue, an organ that is vital for your survival, such as liver and lungs and all that kind of stuff. So effectively, as the growth becomes more, as it develops, it can pick up characteristics that allow it to invade some vascular system to travel through the body. And then once it's got into the vascular system, it can then exit the vascular system into another tissue, and there's a chance that it may be able to colonize and grow. Such as here is showing some cells that have that have grown within the liver from an initial tumor in another part of the body. And so it's this idea of metastasis. It's a breaking away from the initial site, crossing a membrane into the vascular system, traveling the vascular system, and breaking out at a new site to form a new cancer. To do this, it needs to lose its attachment to its neighboring cells, and it needs to digest through the extracellular matrix into the vascular system. It's got to survive the journey, and then it's got to break out of the vascular system into new tissue, which often it's capable of doing. And so the other key thing that we were interested in as well is this idea of angiogenesis, which meaning that tumors require a high degree of blood flow for them to grow. So without increased blood flow to the site of the tumor can't get more than maybe, say, 2 millimeters in diameter, a rough guess. Okay, but if that small tumor 2 millimeters in diameter, can release factors that encourage blood flow to the tumor. Then the increased nutrients allow it to grow to a larger size. So, so effectively, if the tumor is able to secrete hormones, it has a mutation that allows it to secrete hormones or have overactive hormones for some description, it can undergo this process of angiogenesis, and effectively one of the factors it may be able to secrete is VEGF, the vascular endothelial growth factor. And this is a key factor which encourages the growth of blood supply to the site of the small tumor, allowing it to grow to a larger tumor. And also, the fact that it's in close proximity to the blood supply allows it to escape into the blood supply and invade more tissues. All right, so I hope that's helpful as a summary of some of the key points from week 2. Okay, thank you.